Hello students, this lecture covers nutrition for Biology 1407, animal nutrition. When we're looking at animals, those animals have digestive organs, and the two choices are for some of the simpler, particularly radial and a few of the bilateral animals, a gastrovascular cavity, which we've spoken of before. And this consists of a single compartment with one entry and exit, a single entry exit. And this is the simplest digestive system, and it processes foods in batches, ingestion, mechanical and chemical digestion, absorption, and then excretion happen as a session, and then it can repeat that process. The other, far more common and much more efficient system is an alimentary canal, which is a complete digestive system. Food comes in through our mouth. There are various regions that specialize in different steps of the digestive process, and then wastes exit through the anus. And this is a continual digestive system. You can continuously eat, continuously digest, absorb, and excrete. Couple of examples right here. So we've talked quite a lot about jellyfish and other cnidarians, and they have this single compartment with a single entry and exit right here. On the other hand, a complete digestive system, the very simplest one right here, which is very, very early vertebrate in this particular case right here, has an entry and a number of specialized regions right here, and then finally an exit point at the end, classically a mouth, some sort of tube leading to the stomach, which would be the pharynx, a stomach and or intestines, and then an exit through an anus. And here's an example of an earthworm, which is an annelid, and it has a complete digestive system running all the way through it. There it is right there in cross section. All right, now also for all animals, but this applies more to the more complex animals with complete digestive systems. You'll find animals that are herbivorous and animals that are carnivorous, and then finally animals that are omnivorous, in which case they do both. Herbivores very clearly use plants as their primary food source. Carnivores primarily eat animals. And then omnivores, of course, can switch between either type of food or a combination of both. There's some things to think about here. Herbivores, the reason they're so abundant is because there's a terrific amount of food available. It's plentiful and widely distributed and doesn't get away from you. On a downside, one of the difficulties of being an herbivore is the plant matter, because of the cellulose surrounding plant cells, is really quite difficult to digest for most animals. And additionally, nutritionally less complete, particularly in the way of amino acids and certain lipids. Herbivorous animals usually have anatomical adaptations that make them better at eating plant matter, and physiological adaptations to take advantage of that anatomy. Carnivores, the big advantage for them is that their food is nutritionally complete. The amino acids and lipids in particular are the ones that are needed by an animal to make an animal. So it's really eating everything that's required to make another copy of itself. But one of the difficulties, of course, is that the prey animals often don't want to be captured and eaten. So they're dangerous to capture, difficult to capture. Again, there's going to be all kinds of specializations that make carnivores more effective. Quite a few animals are omnivorous to various degrees, including us, and we are able to eat a wide variety of foods, but we're not quite as good at digesting and consuming either of them or capturing them. We're more of a generalized anatomy that's okay at both tactics. All right, when we're looking at a vertebrate digestive system, it's also referred to as a GI tract, gastrointestinal tract, or alimentary canal, all mean the same thing. So y'all know this stuff already. There's a mouth, there's an esophagus, which is a tube, a muscular tube leading to a storage container, the stomach, and then a winding set of intestines, small intestines, a receiving chamber called a cecum, and then large intestines, and then finally an anus at the end of the system right here. This has been terrifically simplified, but it gets the major components of it. Now here's an example of the same, what's called monogastric, which means one stomach, system that's specialized for an herbivorous animal, like a rabbit in this particular case. And one of the things we see is that the intestines are quite a bit longer. That's to handle the more difficult task of digesting plant matter. In the case of the rabbit and other rodents, they have a very, very large cecum, which is a storage organ right here. There's a certain amount of fermenting activity occurring in this region right here. And then the large intestine, posterior to the cecum, and then finally the anus at the end. The large intestine is also often referred to as the colon. All right. Here we have some examples of different multi-chambered digestive systems right here. This one's a bird, an herbivorous bird. This is a chicken, if you recognize that head right there. And this is a goat right here. And both of these animals have a multi-chambered digestive system, which is specialized for dealing with difficult to break apart food. In the case of the bird, there's a crop, all right, then a gizzard. And these are components, these are modified parts, if you will, of the stomach right here, and then an extensive digestive tract after that, the small and the large intestines. Large intestine relatively short in this particular case, and then finally the posterior exit. Birds and reptiles have a combined exit for everything. Their reproductive, urinary, and digestive systems, and that structure is called the cloaca. This is a ruminant, a goat, sheep, horses, cows, all fit into this group right here. And they're noted by having a multi-chambered stomach right here, where food is passed back and forth through a series of different chambers, regurgitated back up here for secondary chewing, swallowed back down and processed, and then finally goes through the intestinal tract, small intestines, large intestines, and the waste excreted. And the purpose of both of these systems, the crop and gizzard system of so many birds and reptiles, especially herbivorous reptiles, and the multiple stomach chambers of a ruminant mammal, is for the digestion of difficult plant matter, cellulose-rich plant matter. And in both cases, the modifications are there to help break apart the cellulose cell walls, gaining access to the contents of the cells. And take advantage, particularly in the case of ruminants, of lots and lots of bacteria living in that digestive system. When we look at mammals in particular, one of the characteristics of mammals is that they have what's called heterodont dentition, as opposed to reptiles, which have different sized teeth, but they all have the same shape in a given type of reptile. In mammals, we can have very differently shaped teeth in different locations for different purposes. So we all, are familiar with our own incisors, canines, premolars, and molars. These are cutting teeth, tearing teeth, cutting and grinding teeth, and specialized grinding teeth. And what we see is that herbivores have a tendency to focus on the grinding and perhaps the cutting teeth. Carnivores tend to specialize on the cutting and the tearing teeth and variations on those themes. So if we look at the dentition of a fossilized skull, we can rapidly ascertain, if it was a mammal anyway, we can rapidly make some hypotheses about the food that animal would have eaten by looking at the dentition. All right. All complex animals with a digestive system have what's called extracellular digestion. So what that means is the food is broken down all the way down mechanically, and then most importantly chemically digested with various digestive enzymes, all the way down to the monomer stage. So, the extracellular digestion ultimately generates a liquid that is rich in single amino acids, monosaccharides, broken apart components of lipids like fatty acids in one case and the glycerol molecules in the second case, and so on and so forth. And then those those monomers, those simple components of macromolecules, are absorbed by the cells of the digestive tract. All right, one of y'all's requirements for this unit is that you do know and are able to enumerate the components of the digestive system of a typical vertebrate, and knowing your own is what you're particularly required to know right here. You need to know the organ itself, what its functions are, a couple of structural details that are relevant to the digestive process, chemicals, enzymatic chemicals that are used by that animal or by that digestive organ. And at the end of the unit, we'll also look a little bit at a hormonal regulation of this process. So just to kind of look at it from an overview here, and this is your digestive system. The digestive system consists of the digestive tract, which is the mouth, esophagus, stomach, small intestines, large intestines, colon, and rectum, and then accessory glands that contribute to that process, specifically the salivary glands. And you've got three pairs, like all mammals do. The parotid glands, the sublingual glands, and the submaxillary glands right here. One on each side. So three on the left, three on the right. Another very important gland that contributes to the digestive process is the liver, primarily as the producer of bile, which is introduced into the digestive system, and it's also stored in the gallbladder. The liver produces bile. It is stored in the gallbladder and introduced into the small intestine when you eat a meal that contains lots and lots of lipids. The final important organ behind the stomach right here is the pancreas, which is the primary producer of extracellular digestive enzymes. The enzymes that do the bulk of the chemical digestion of food are produced by the pancreas organ. All right. Primary function of the mouth, of course, is a pathway in for food. A terrific amount of mechanical digestion through biting and chewing, crushing food down into smaller and smaller bits. The purpose, of course, of mechanical digestion is to reduce the size of the particles of food and increase the surface area available for digestive enzymes to act upon. The more surface area that a given amount of food is converted into or made available for it, the more rapidly it can be chemically digested. Tongue and teeth and cheeks and lips all contribute to manipulating that food as it's ingested, as it's chewed, and then swallowed in the form of a bolus, which is a semi-liquefied pile of food. All right. The pharynx is what you and I would refer to as the throat. And there is the nasopharynx, the oropharynx, and the lingual pharynx, the three parts of the throat right here. And its inferior boundary is the epiglottis, which is where the pharynx divides up into the esophagus, leading down to the stomach, and the trachea, leading to the lungs. And the epiglottis is a valve that controls access to that airway, so we don't accidentally inhale food or liquid. Also, there are some valves in the soft palate, the back of the roof of the mouth, that close off access to the nasopharynx. So when you swallow food, those valves naturally close and generally keep food and liquids from going up into the nasal cavities and force food down into the esophagus. An illustration of that process. So here's the soft palate closing off the airway leading up into the nasal cavities. The epiglottis is pushed downwards and naturally seals off the trachea when food is pushing past it, and liquid to a lesser degree. And the tongue moving backwards also helps close the epiglottis. And so food, the bolus of food, is preferentially pushed into the esophagus. Once we introduce food into the esophagus, we don't have to consciously manipulate it anymore. The esophagus has two layers of smooth muscle. There's longitudinal muscle running in this direction, and then circular muscle going around in loops around this tube right here. And rhythmic contraction of both the longitudinal and the circular muscle creates these segmentary waves of contraction running down the esophagus that push boluses of food down all the way to the stomach, and that is called peristalsis. And that process, with some modification, occurs all the way through the digestive tract. All right, the first organ is going to be the stomach, and the stomach has two big roles. One is storage of food, so that you can eat a lot of food in one go and store it in this highly variable chamber. You can fill it with food, and the walls stretch tremendously, so it can increase its volume quite a lot. And then also quite a bit of mechanical and chemical digestion occurs in the stomach right here. I did forget. I need to go back very quickly right here. When we are chewing food, we introduce saliva into that food, which is mostly water and mucus for the purposes of lubrication to allow food to be swallowed more easily. But we also introduce the first of several enzymes into the chewed up food. The most notable one that has the biggest effect is salivary amylase, which breaks starch down. So if we chew a starchy material for quite a long time in the mouth right here, after a while the action of amylase liberates individual glucose molecules which we can taste. We can't really taste starch, but we can taste glucose, so the food will start tasting sweet if it's starchy, given time, due to the effect of salivary amylase. All right, once we get into the stomach, we release some other digestive enzymes. The most notable one, and the one you need to know, is pepsin, which is an enzyme that breaks apart long polymers of amino acids into shorter chains. So it breaks proteins partially apart. Additionally, the stomach has acid, hydrochloric acid, which it produces. We secrete HCl right here. And that acid lowers the pH dramatically in here, which tends to denature proteins. So the acids denature the proteins, so they unfold. The pepsin gets at bonds in the polypeptide chain and breaks them apart. And that is the major digestive activity of the stomach. So the stomach is aimed at mechanical digestion, that through muscular contractions and mashing and squishing of the food right here. And then chemical digestion by the combined action of hydrochloric acid and pepsin, targeting primarily proteins. Then we move that digested material into the small intestine. The proper name for the liquid coming out of the stomach is chyme, C-H-Y-M-E. That is the acidic, highly liquefied remains of food at this point. There's a sphincter that controls movement from the stomach up into the esophagus. That's called the cardiac sphincter, and then the lower sphincter is called the pyloric sphincter, which controls movement of food from the stomach to the small intestines. The small intestine. You do need to know this piece right here. The first portion of the small intestine is referred to as the duodenum. That's the very first loop. And that's an important regulatory and introductory component of the intestines, the duodenum, first loop of the small intestines. It's at this point that secretions from the liver, which of course is bile, and secretions from the pancreas, both are introduced into the small intestines. So as food, as chyme comes out of the stomach and goes into the small intestines, various amounts of liver and pancreatic secretions are introduced into that stream. The role of bile is to digest, or not to digest, but to emulsify fat droplets. So if you have quite a lot of lipids in your diet, in the food that's coming out of the stomach right here, the liver via the gallbladder will release quite a bit of bile, which acts like a soap to break up that fat and make it soluble in water. The process is referred to as emulsification. All right. Here's an illustration of the liver, which has a huge number of metabolic roles. But in this particular conversation, the key thing is that it extracts materials from the blood that are actually left over from the recovery of hemoglobin from dead red blood cells. And the heme group is combined with some other chemical structures to create the material known as bile, which is collected together in ducts, ultimately the bile ducts of the liver, which fill the gallbladder, which is simply a muscular storage organ, a little reservoir. And then if a whole lot of lipids are introduced into the duodenum, quite a bit of bile is ejected into the duodenum to emulsify those lipids. And then here's the pancreas, which produces hydrolytic enzymes that break down proteins, carbohydrates, lipids, and nucleic acids. So all the macromolecules have enzymes that target them that are produced by the pancreas. All right. Three other enzymes that we want to identify that you'll be required to know for examination purposes are produced by the pancreas. All right. So it's referred to as an exocrine gland. That means it's a gland that releases liquids that can find a way to the outside of the body. Some others are the salivary glands, sweat glands, and mammary glands. They're all exocrine glands. And again, the pancreas dumps its liquids into the duodenum. And three target enzymes you want to know about are pancreatic amylase, which digests starch to make a disaccharide called maltose. Trypsin, which breaks proteins down into very short peptide chains, usually dipeptides. And then lipase, which takes triglycerides and breaks them apart into glycerol and the individual fatty acids. All right. And then finally, the epithelial cells of the small intestine produce enzymes that will break this disaccharide maltose into its two-component monomers, take any remaining di and tripeptides and break them down into single amino acids. And that's the final step of chemical digestion that occurs in the small intestines right here. And at the same time, we'll be doing absorption of materials. All right, this slide shows just a summary of the entire process of digestion and absorption. So the terms mechanical digestion and chemical digestion should be fairly obvious. This is, the same sort of thing that would happen if you mashed and crushed and smashed things, chewing in your mouth. Churning in the stomach. Your stomach does quite a bit of mechanical digestion. And your small intestines to a lesser degree as well. They spread that liquefied pasty stuff out and rub it back and forth, spreading it out over a wider and wider surface area. So that's all mechanical digestion, which gives more surface area for the enzymes involved in chemical digestion to do their work. Remember again that enzymes are chemicals that catalyze a chemical reaction, are not used up in that reaction, and so are available to participate in another round of that reaction right there. So those enzymes are used for an extended period. They're present all the way through here and continue acting. Now, things that salivary amylase, for instance, is denatured and made inactive by the low pH of the stomach, so it doesn't make it out of the stomach intact. But the enzymes that are released in the duodenum from the pancreas act on food all the way through this process here until there's really nothing left. So mechanical and chemical digestion both start in the oral cavity and proceed all the way through the system right here, with initially the focus being heavily on mechanical digestion and the emphasis later on being increasingly on chemical digestion as we work our way towards the end right here. And then we've always got peristaltic muscular contractions that push food down the esophagus, around in the stomach right here. 2 layers of muscle, 3 layers of smooth muscle, 2 layers of muscle, longitudinal and circular muscle surrounding the tube all the way down through the end right here. And then, of course, most importantly, the goal of all of this is to absorb the nutrients out of here. So what are we going to pull out? Well, the monomers, the monosaccharides, the single peptides, the single amino acids, the components of the lipids, the single nucleic acids are all absorbed by cells, primarily in the small intestine. Water and salts are primarily absorbed by cells in the large intestine. As we get towards the end right here, we increasingly absorb water and salt. We introduce a lot of water in order to aid in digestion. Lots of water makes the chemistry work better. And then we attempt to recover as much of that water as we can in the end right here so that we retain the water, reabsorb the salts. Salts are used to move water. Introduce a lot of sodium right here. A lot of water follows by osmosis. Absorb a lot of sodium right here. Water follows by osmosis and is pulled back out. A number of diseases that disrupt that osmotic regulation in the large intestines are collectively referred to as diarrhea, and which can be, in extreme cases, life-threatening if too much water is lost through the waste stream of feces. All right, and what's left is feces, and that is expelled out the anus. And feces is simply mucus, a lot of bacteria that live in the small large intestines right here, and the indigestible material, what was not really an absorbable amino acid, what was plant fiber that we didn't successfully break down, all of those sorts of things. Whatever is not a monomer is left behind in this pile of stuff referred to as feces. So it is not waste products from your body that are introduced into the system. Feces is simply what cannot be assimilated. Absorption, another word for absorption is assimilation, to pull that stuff out of your food and take it into your body and use it. What can't be assimilated is left in the digestive tract and excreted as feces. All right. This just covers those ideas again right here. A characteristic of the large intestine of all organisms is that there's usually quite a lot of, where does it say it right here? There's quite a bit of bacterial digestion that occurs in this area, and a lot of secondary metabolic processing by bacteria that occurs in this region right here, including in us. We have our E. coli that naturally live in here, along with several other strains of bacteria that live in here, and break down a lot of the indigestible material that makes it to the large intestine for their own purposes, and they produce substances that we then can absorb, that are breakdown products of that food, and including the final versions of a lot of mineral molecules that end up being vitamins for us are liberated by bacterial digestion in the large intestines right here. So this is a form of bacterial fermentation occurring in the large intestines. All right, a characteristic of digestive tracts that kind of scales up with the metabolic activity of the animal, the size of the animal, and how much it does, is adaptations that increase the surface area of the digestive system. We've talked about this quite a lot. So the characteristics are the fact that, I'll go back up here, the digestive system itself consists of a very long, highly folded tube packed into a small body cavity. There's an adaptation for increased surface area. And then we have lots of ridges and furrows on that surface, on the surface of the stomach and the intestines, which increase their surface area. Here's an example. Let me go back to the stomach. These ridges right here called rugae on the stomach. And then the inner wall of the stomach and the small intestines is folded up into large multicellular structures called villi, which dramatically increase the surface area of these organs. And then finally, those villi, the cells on the surfaces, have terrific cellular extensions folded back and forth, called this image right here shows a single sort of stylized epithelial cell lining the small intestines, and these would be the microvilli characterizing the surface of that cell. So this is that's a very microscopic structure, and then those cells in turn are organized into larger structures called villi that line the inside of the digestive tract. All right. And so those cells together collectively organized into these larger structures of villi. Microvillus, villi, and then rugae and folds and things like that. All right, what else? Okay, and of course on the other side of that epithelium, which is typical of all epithelia and certainly important right here, we have a terrific amount of capillaries bringing the blood supply and the lymphatic system for that matter in close contact with that epithelium, which is absorbing these nutrients from the food stream. All right. A little bit about types of digestion right here. I'm sorry, got a little ahead of myself right there. So carbohydrate digestion, we want to talk about that a little bit. Remember, we have polysaccharides. We take in a lot of our carbohydrates in the form of starch. So we have amylase in the mouth that breaks starch down into shorter carbohydrate chains. We have maltase, an enzyme that's released by the lining of the small intestines that takes maltose and breaks that down into glucose, which can finally be absorbed. We also have pancreatic amylase released by the pancreas that digests what's left of starch into maltose. Sucrose is broken apart by sucrase into glucose and fructose, which can each be absorbed. And lactose is broken apart by lactase. In mammals, and typically only in infant mammals, but in some adult mammals like some humans, lactose can still be digested and absorbed. Cats and dogs cannot. If you feed your cat lactose, it cannot digest that lactose and break it down into glucose or galactose, unless it's a kitten, in which case it produces lactase. All right. Protein is fairly hard to digest and requires the function of a very dedicated organ, the stomach, to really do that process properly. In the stomach, we have pepsin that's released, which begins the digestion process in concert with the low pH caused by hydrochloric acid, which denatures the proteins. That allows pepsin to get at the bonds that it breaks and breaks those proteins down into short polypeptide chains. Then in the duodenum, you've got trypsin, elastase, chymotrypsin right here, which are all specialists that work at either one end or the other end or the middle of a short polypeptide chain and break those things down until we get down to very small peptide chains. A few more right here that finish off that process of digestion so that the result is free amino acids that can be absorbed into the bloodstream by the cells of the small intestine, the epithelial cells right there. So trypsin, elastase, chymotrypsin, carboxypeptidase, dipeptidase, aminopeptidase are all digestive enzymes targeting proteins. Lipid digestion is not quite so complicated, but it does require two components. First, you must have that bile, which acts as a emulsifier, making the lipids partially soluble in water so that the enzymes that break the triglycerides apart can get a hold of them. And then we have lipase, primarily lipase, which is the enzyme that breaks the fatty acids away from the glycerol molecule, breaks those triglycerides down. That enzyme is referred to as lipase, produced by the pancreas, and also versions of it produced by the epithelial cells of the small intestines, lipase. All right, we've already looked at that. So when we get to the end of the digestive system, again, we get into the large intestine, which consists of a number of sections. The most distinct bit is the cecum, which is a slightly enlarged region that is the junction between the small and large intestines. And then the various, permutations of the colon. And then finally a collecting and drying, desiccating region called the rectum, and the anus at the end. All right. In humans, the cecum has a bit of an extension, a very small extension called the vermiform appendix. This is the appendix that is occasionally of medical note because it becomes inflamed and ruptures, causing a septic inflammation of the tissues in the digestive cavity right here. All right.